Any questions?
We have a test now.
I know we do. We're a week from Thursday, right? Week from Thursday. Fair amount of inoculating tomorrow of your unknown. We'll work in the same groups that we worked in last time to kind of minimize all the controls you have to do. So make sure you have a slant that's got a decent amount of viable growth on it. Doesn't have to be any particular age. And you'll save yourself some time if you read ahead and figure out what we're doing.
Focus.
Okay, we were in the middle, or just the beginning, sorry, of talking about some of the overview of the immune system. And we divided the immune system into 3 lines. One of the things that I do want you to understand is that these lines are not hard and fast. And sometimes there are overlaps between them. For example, there are cells that might participate both in line 2 defense and line 3 defense. There are some chemicals that might be involved in line 1 and line 2. So the distinction between those is a little bit blurry, but that's okay. It gives you a general idea. Which of the 2 lines are considered to be innate immunity? 1 and 2. Those are the ones you are born with. Line 3 is adaptive. Okay? What are the major players in line 3? Antibody-producing cells. Those are lymphocytes, and we said there were 2 kinds of lymphocytes. What were they? B and T. Okay. There's another player I'm not sure that I mentioned. Did we mention macrophages? Okay. Macrophages, just start introducing you to some of the, some of the players. That macrophages are involved in helping an interaction between the lymphocytes and help to facilitate a communication between those lymphocytes. So we'll talk a lot more about macrophages. This is kind of big picture right now. The 3rd line of defense is traditionally divided into 2 branches. Humoral immunity and cell-mediated immunity, or CMI. Now, the major players in immunity are the B and T lymphocytes. Does anybody know which of those 2 are responsible for humoral immunity? B, yes. Because humoral immunity involves the part of the immune system where antibody is produced. The word humoral doesn't have anything to do with being fun. It's a Latin word for fluid. The body's humors are fluids. And these antibodies are typically secreted into the body's fluid or circulatory system. Typically, this response is mounted against pathogens or antigens on the pathogens that are soluble. They're floating around in your body's fluids. Maybe that's blood, maybe that's the lymphatic fluid. But they typically haven't invaded a cell yet. Okay? So what the antibodies are trying to do is they're trying to neutralize these things before they can infect cells. Now, as it turns out, there is a type of T lymphocyte that is oftentimes involved in humoral immunity as well. That T helper cell oftentimes, but not always, is needed to kick the B lymphocytes into activity so they can start producing this protective antibody. So I guess technically both T and B lymphocytes are involved in at least some aspects of humoral immunity. Cell-mediated immunity. This is really the province of the T lymphocytes. T helpers also play a role here. in that they activate cells, these immature T-naught cells, which don't really quite have an identity yet. And one of the things they activate them into is T cytotoxic, or some people like to call them T killer cells. These T killer cells attack your cells. But they only attack your cells if your immune system's working properly, if they have become infected by a pathogen, or if they are cancers. So these kill host cells that are infected / cancers. Remember that I said the immune system had some redundancy built into it? Okay, well this is an example, okay? Because there is a system that kills cells that have already been damaged. But then there's also a system that tries to prevent that damage from occurring, the humoral immune system. If something gets past the humoral immune system, then it should be taken care of by the cell-mediated immunity, T cytotoxic guys. Notice the central role that T helper cells play in immunity. This is why HIV is so devastating to the immune system. Because the HIV virus specifically binds to and infect T helper cells, and consequently wipe out both branches of your immune system. There are some genetic defects that are B cell defects. Can you survive with a disabled B cell response, with a disabled humoral immune response? Yeah, but it makes you pretty prone to infection, but you still get your cell-mediated immunity. There are certain defects genetically that wipe out this branch, but you've still got your humoral immunity. But if you lose both of these branches of immunity, or they're severely depressed because your T helper cell population is really plummeted because they're being killed by this HIV virus, that's a real problem. One of the keys to the immune system is surveillance. You have within your body populations of white blood cells which can move around and crawl around within your body. They move kind of like an amoeba does, if you've ever seen an amoeba move. In fact, we call it amoeboid movement. And these cells are stationed at a lot of key points in your body, particularly ones that might be common portals of entry for pathogens. So the skin, or the mucosal tissue associated with your gut or your respiratory tract. There are these populations of these cells that are involved in surveillance. And they go around, and they are basically the dirty old men of the immune system. Because they grow all of the cells. And they check them out. What's happening is there is cell-cell recognition going on, involving receptors on one cell, receptors on another cell. And if these cells detect something that shouldn't be there, it's foreign, then they take action. And that action might be chewing it up and spitting it out, phagocytosis. But oftentimes what they do is they ingest these things, they display pieces of that pathogen on their surface, and then they migrate back to the lymphatic tissue, like your lymph nodes, and show that to other immune cells and say, hey, look what I found. And when they do that, that is part of this activation that occurs that turns on T helper cells and B cells. So these antigen-presenting cells which include macrophages and dendritic cells, play a really important role in the immune system.
By detection, do you mean like the detection of like the capsule, the virus and whatnot?
It's usually not the whole capsule, but it might be certain key molecules or even portions of molecules that are part of that capsule. We call them epitopes or antigenic determinants, and we'll get into that in more detail later on. But yeah, they're responding to substances that the host cells don't have, but that the pathogen does have.
What kind of molecule would that be?
Pardon me?
What kind
Oh,
of-
it could be a capsular protein, could be some DNA. Proteins are huge antigens. Okay. Oh, that question's back. Okay, so that's kind of our overall big picture. So here's another slide that just kind of shows you the same thing in a different way. I like in this particular section just to kind of give you different ways to conceptualize this. Some people like these kind of branching diagrams right here. Other people like some of the other things that we've seen. But again, this breaks down into innate and acquired, first, second, and third line, and it gives you some ideas of what some of the components are in each of those lines of defense. So we're going to start over here, first line of defense. Talking about physical barriers, chemical barriers, and genetic considerations. Some of this we've already talked about a little bit, and so I will refer back to it. Particularly the physical barriers, because we talked a little bit about some of those when we talked about bacteria overcoming those, what their virulence factors were. So, we already talked about the skin as a physical barrier. Your skin is a pretty remarkable organ. It is constantly renewing itself. It is constantly shedding. And that removes pathogens from the surface of your skin. Of course, they just recolonize. You shed about a half a pound of skin per year. That's what a lot of dust is in houses, is dead skin cells. And then you got little ecosystems of, little mites and stuff that feed on that dead skin. Some people are allergic to them, otherwise they don't really harm you. That's about 10 billion cells per day of skin that you shed. It's quite a bit. Skin cells can secrete some antimicrobial substances, some peptides. Those particular antimicrobial peptides are called defensins. There are some cells that are associated with the skin that are involved in surveillance. The dendritic cells are commonly patrolling in the lower layer of your skin, looking for pathogens to engulf, and then going back to the lymph nodes and other parts of the lymphatic system and alerting other immune cells that they found something wrong.
This would be a good question, but dendritic cells are not associated with like brains.
No, they have long, it's a good question though, they have long protruding branches. I'll show you a picture of one in a second of their cytoplasm, and they look kind of like that projection of the dendrite on a neuron. Okay, I think dendrite literally means branch. Yeah. The skin does have openings that are potentially infected. We saw some of the staph infections that you could get if you got infected hair follicles, for example. But there are some adaptations that help to prevent that infection from occurring. Sweating is one of them. So as sweat comes out of your skin, that tends to wash the pathogens out of those little pockets in the hair follicles. Sweat is a little bit basic, and so that change in pH is inhibitory to some microorganisms. Lysozyme The enzyme that breaks down peptidoglycan can be found in sweat. So that's part of your protective mechanism. Sebum is a fatty substance. It's produced by the sebaceous glands in your skin. The sebum makes the skin more pliable, so it's not dry and brittle. If your skin gets all dry and brittle, it's more likely to crack and break, therefore compromising that defense barrier. And sebum has some fatty acids which help to lower the pH, and that is inhibitory to some microorganisms. The skin really is a pretty great barrier. And unless it's compromised, it really is pretty good about keeping out infectious agents. That's why burn victims are in so much trouble, especially for burns from acid. Because if you compromise a huge portion of this defense barrier, usually what burn victims die from is infection. This can't be controlled.
But it can be rejuvenated, yeah?
Skin, yeah. Yep, yeah, and if they can, if it's not too great, they can do transplants. But, yep. It's just a matter of fact, if you can repair the barrier, while the infection's going on.
Do scars form the same way? Like, does that make sense?
Yeah, well, scars, to completely regenerate skin, stem cells really have to be involved, because they didn't have to differentiate into all the various tissues of the skin. If tissue is injured and it's in an area where there isn't really good stem cell involvement, then the cells start secreting collagen and other things and kind of just fill in the gap. And that's far less pliable and much more likely to crack and be compromised. And so areas of scarring are potentially areas where future infection could take place. Your GI tract is another portion of your body that could be prone to infection, as is your respiratory tract. So again, those cells, we talked about the mucus secretion, we talked about peristalsis in the GI tract, flushing things out of your gut. We talked about the ciliary escalator, where those cilia in your trachea, for example, sweep things up out of your respiratory tract. There's also some chemicals that are produced by those cells. They also produce those defensin molecules. They also can produce some lysozyme. And then we've also got a pretty robust population of dendritic cells in those epithelial tissues of the mucosa. And what they can do is they can kind of crawl across out of the blood that cellular barrier, and they can stick one of those little dendrites out and kind of sample the area and see if there's some pathogens there. And then if they find them, they can grab them, pull them back in, ingest them, and then run off to the lymph nodes and say, got an infection, got an infection. One of the books I was reading said it's kind of like a submarine poking its periscope up through the surface of the water. You kind of poke one of those dendrites through that epithelial barrier of the gut and try to see if there are any pathogens right on the surface there that have attached, and then grab those and take care of them. So a lot of interesting things are going on within that skin and mucous membranes. Your GI tract also secretes some chemicals which are inhibitory to certain microorganisms. There is a substance produced by the gallbladder called bile salts. And we're actually going to use a media in lab later on that has bile salts in it. And those bile salts tend to inhibit selectively the growth of gram-positive bacteria. So that's kind of good, because some of those nasty pathogens, like all those Clostridium, they're gram-positive. But some of your normal flora, the enterics, are gram-negative, so the bile salts don't inhibit those. Stomach acid destroys a lot of microorganisms. One of the reasons that you produce mucus is to coat bacteria that get into your respiratory tract. You cough it up, and if you don't hack it out, you might swallow it. And so that slime with the bacteria in it drops down into a vat of acid and pretty effectively kills those microorganisms. That's really the primary role of acid in the stomach, is to destroy microorganisms. There's really not much hydrolysis of food that occurs in the stomach. Remember, most of that happens in the small intestine. Okay. We talked a little bit about iron and the role that iron plays in microbial metabolism. And there is oftentimes a situation in the body where iron is really a limiting factor for microbial growth. No iron, and you can't make things like cytochromes, for example, which are a key part of your electron transport system. Actually, iron is very insoluble in the blood. And so humans produce a protein, a blood protein called transferrin, that binds iron and helps it to be distributed throughout the body. The pathogen Staph aureus can produce a substance called a siderophore. It's another protein. And it actually has a higher affinity for iron than transferrin does. So it kind of outcompetes the transferrin. Well, the body sometimes responds by producing another protein called lactoferritin. And that can grab the iron from the siderophores. So it's really a tug of war over iron that's happening a lot of times. And then I think I mentioned this with the hemolysis. Some bacteria just go, heck with this, I'm just going to hemolyze some red blood cells and grab my iron there.
Why would the body just make that all the time?
Don't know. So here's kind of a summary slide of some of the kinds of activities and defenses that contribute to the first line. Saliva, stomach acid, another iron sequestering molecule called gastroferritin, didn't mention that one. It's kind of like transfer or lactoferritin. Bile we talked about, some intestinal secretions, peristalsis, vomiting, urinary system, urine contains lysozyme, prostate secretions, transferrin. So this kind of gives you a fairly exhaustive list of some of the kinds of things that are happening with that first line of defense. Now, in addition to physical barriers and chemicals, there are some genetic considerations that we need to talk about briefly regarding the first line of defense. There are some well-documented racial and ethnic differences in susceptibility to certain diseases. Part of this has to do with where the disease originated and how long as a race you have been exposed to that particular pathogen. Because what's going to happen over time? Natural selection is going to occur, right? And if random mutations occur that result in resistance to that particular disease, the disease will select for individuals that become resistant to that particular infection. So smallpox, for example, we believe was European in origin. That's where that disease arose. It was not indigenous to North America. So Native Americans had never been exposed to smallpox until Europeans settled the North American continent. And now all of a sudden, people who were carriers and resistant came over and exposed the Native Americans to these particular diseases. And they had no really innate immunity to it. And therefore, they were much more susceptible to that disease. Unfortunately, I think it's fairly well documented that when we were colonizing this country, and especially when there was the attempt to wipe out the Indian Native American population so we could take over, that actually bioterrorism was practiced on the Indians by distributing smallpox-infected blankets to the Indians. There's a lot of evidence that indicates that that really did happen, and that's a pretty dark period in American history for that to have happened. Mostly these racial and ethnic differences are the result of lack of virus receptors for that virus to attach to the cell, or modified receptors that the virus can never recognize. So modified or lacking viral receptors. There is oftentimes a species barrier between viral infections. And for example, avian or bird viruses oftentimes cannot infect humans. Why? Because their receptors match up with bird cells, but not with human cells. But unfortunately, those viral receptors mutate very rapidly. And if the right mutation occurs, and now an avian virus can recognize a receptor on a human cell, then that virus has been able to jump that species barrier. And that's what's of real concern with influenza outbreaks, for example, because many of those are avian in origin. So like the H1N1 virus, the bird flu virus.
These receptors that you're talking about, it's like on the host cell,
Yes.
right? Okay.
Yep.
So they don't have risk.
Well, I mean, viruses have molecules sticking out of their surface which can
Right.
mutate also, which bind to those receptors, yeah.
Okay.
Yeah, but when we're talking about racial and ethnical differences, we're talking about human host cell receptors on cell surfaces. Pleiotropic genes are something we should mention when we talk about susceptibility to infection. I remember what pleiotropic genes are. We all had genetics, right?
Yeah.
Yeah.
Isn't it when one gene affects like multiple phenotypes?
One gene affects multiple phenotypes, and what is kind of the classic example of pleiotropism? Sickle cell? Remember sickle cell?
Yeah.
Yep. And what does being heterozygous for the sickle cell gene give you resistance to? Malaria. Yep, coming up in one of the seminar talks, right? Who's doing that?
I'm doing malaria.
You're doing malaria.
Yeah, no, I would have mentioned sickle cell.
One of the most devastating genetic impacts on the immune system are immune system defects that have a genetic origin. And one of the more interesting ones, but more tragic ones as well, is an immune condition called SCIDS. You know what SCIDS stands for? Severe combined immunodeficiency syndrome. This is due to a single gene defect in a key enzyme, adenosine deaminase. And if you have a defective ADA gene and you can't produce this enzyme, it interferes and inhibits the production of both B and T lymphocytes. So basically you have no immune system. There was a little kid named David in the 80s, I believe, that was diagnosed with this prior to birth. And they had to transplant him into a plastic bubble, which basically isolated him from all contact. That was actually one of John Travolta's first movies. He played David and the boy in the plastic bubble. Okay. They actually have had minor success in treating this particular condition through replacement of this particular protein by using a virus to deliver a good copy of that gene to stem cells in the affected individuals. And there actually were a couple of people that were, I believe, cured by this particular technology. But they started running into problems with people dying from viral infections because the viral carrier was causing cancers and things like that, but they hadn't anticipated. So I think they pretty much shut down those particular trials. I'm not aware of them doing anymore. But there was a little bit success of trying to correct this genetic defect at the gene level by replacing a good copy, bad copy of the gene with a good copy.
So similar to like Ebola vaccines, or treatments, I don't remember. They use like, I don't know, viruses from like chimpanzees and stuff to like deliver DNA from like Ebola. Can those potentially like cause cancer?
I mean, theoretically, yes, but that's all part of the clinical trial process, is to try to determine whether that does indeed happen. Yeah. I mean, anytime we really introduce a virus, and particularly if that virus is going to insert genetic material into your genome, if you can't have absolute control of where that insertion takes place, then you could alter gene expression. So it's not without risk. It's not without risk. Okay, now we're going to step back for a second before we hop into second line and do another kind of big picture of the immune system. But instead of doing it from kind of a functional point of view, we'll do it from kind of a structural point of view. So let's take a look at the overall structure of the immune system. Where is your immune system in your body? Yeah, it's all over, okay? We're going to talk about it in terms of 4 body compartments that are all interconnected with one another. So if something is introduced to the immune system in one compartment, it eventually will be transferred to the other compartments as well, because they're all interconnected. Anybody have any idea what compartments I'm talking about? Blood is one of the compartments, yes, and that's one we'll spend quite a bit of time talking about.
Tissue?
Pardon me?
Tissue?
Yeah, the extracellular fluid.
Is lymph separate from that?
Nope, lymph is another one. Unless you've been reading ahead, you're not going to know the 4th one. I'm sure you've never heard of this before. The RES system RES stands for the reticuloendothelial system. So we're going to knock off RES and extracellular fluids pretty quickly. I think you should know about them, kind of have a brief idea of what they're doing. But we're going to spend most of our time on the blood and the lymph. Okay, so what is the reticuloendothelial system? It is a system of fibers that emanate from cells called the reticular fibers. And they project from one cell to another, and they're very common in connective tissue. And they, in some respects, kind of help to stabilize and hold that connective tissue together. So we've got this extracellular fluid that bathes the cells. Then we've got these cells, and then we've got these reticular fibers that kind of connect the cells and emanate out from the surface of one cell and contact another one. And these are some of the parts of the body where the reticuloendothelial system is very pervasive. There's a lot of this particular kind of tissue. Now, why is this important in terms of the immune system? Well, this is home to a lot of white blood cells, and particularly macrophages and dendritic cells. I like to kind of refer to these two cells as cousins. They have a lot of similarities, but they're derived from a little bit different familial line in cellular development. And they have a little bit different functions, but they're very closely related otherwise. So we've got these cells, these white blood cells, that can move throughout the body. And they kind of crawl in between the cells of the tissues along these reticular fibers. It's almost kind of like they're tracks, and they're motoring along on those particular tracks. And what are they doing? They're involved in surveillance. So they're out there checking out cells, looking at their surfaces and determining belong, don't belong. These particular cells are of two types. We designate them either wandering or fixed. And the wandering cells really do just kind of continue to move around from place to place. The fixed can still move, but they tend to reside in particular locations within the body. And those oftentimes are locations where there are really potential for infections developing in those particular locations. These fixed ones are called histiocytes because they're associated with particular tissues of the body. Histology is study of tissues. So, for example, you have a population of histiocytes called alveolar macrophages. And these obviously are there to help protect your respiratory system, associated with the lung alveoli. Think about all the potential for inhaling particles and then getting into the alveoli. So it's good to have some protection there. There are Kupffer cells that tend to localize in the liver. There are Langerhans cells. Whoops. That tend to be associated with the skin.
If they're fixed, how do they take them to the, do they become unfixed afterwards?
They're still able to move. but they just have a tendency to find a location and then just hang around that one particular area.
Okay.
Okay? Whereas these wandering ones are just kind of going, everywhere. There are glial cells in the central nervous system. So this gives you a little bit better idea of kind of what's going on in this extracellular fluid and in the reticuloendothelial system. So here's a blood capillary, okay? And if there are infectious agents in the blood, there is typically some leakage of fluid out of the blood into the surrounding tissues. And sometimes, in part of your immune response, that leakage becomes really pronounced. And fluids actually intentionally rush out of the blood into the surrounding tissues. And we get swelling or edema as a result of that. That's part of inflammation. Okay? Well, pathogens can then be thrown out into this extracellular fluid, or maybe they were introduced there by a break in the skin, and you get an infection in that particular skin tissue. So then you've got pathogens in the extracellular fluid. Well, there are these cells patrolling within the reticuloendothelial system that deal with these. And also, whatever is in this extracellular fluid ultimately is absorbed by the lymphatic system. Remember, that's one of the purposes of the lymphatic system, is to drain off fluid that results when we get leakage of fluid out of the circulatory system. If we didn't have a lymphatic system, once you swelled, you would never go down again. Okay? So then some of these pathogens are carried through the lymphatic system. We'll talk about lymph nodes and stuff later on, but eventually they get to lymph nodes where there's a whole bunch of white blood cells waiting for them. So the connection between the blood, the RES system, including the extracellular fluid, and the lymphatic system is really important in terms of keeping this whole system connected and coordinated. Okay, lymphatic system. One of the purposes of the lymphatic system is just what I mentioned, to return fluid that is leaked out of the circulatory system into the tissues back to the circulatory system again. Second purpose of the lymphatic system is to transport fats to the circulatory system. Now remember, when most things are digested in the small intestine, so for example, when proteins are hydrolyzed into amino acids, or carbohydrates are hydrolyzed into sugars, those smaller molecules can then cross the intestinal wall and be absorbed directly into the circulatory system and then transported to the tissues. But fats cannot. Fats instead are absorbed by little projections of the lymphatic system. Remember what those little projections are called? Lacteals. And then the fats travel through the lymphatic system first, and then ultimately the lymphatic system empties into the circulatory system, thoracic duct. So that's why lymph has kind of that fatty, yellow, glistening appearance, is that there's fat in it. the part of the lymphatic system we're really going to be interested in is its role in the immune system. This is where most of the interactions take place between pathogen and defender. Now that might seem kind of funny, and it might seem a little bit counterproductive. Think about it for a second. If you have a break in your skin and you get an infection in your skin, yes, there will be some activity occurring by some parts of your immune system to deal with that infection right at the site where it occurred. But a lot of it's going to involve those wandering dendritic cells grabbing pieces of that pathogen, displaying them, and carrying them back to the lymphatic system, the lymph nodes, and they're then activating those lymph nodes, cells, the B and T lymphocytes. And then eventually those pathogens are going to be carried by the lymphatic system to those activated B and T lymphocytes, and that's really where the most of the destruction actually ends up taking place. So one of the things that happens during infection is we get a real expansion in the number of lymphocytes, because when they get the proper signal, these cells start multiplying and they form clones of themselves. It's called clonal expansion. And the numbers of white blood cells just spikes tremendously in those lymph nodes. So what's the doctor looking for when they palpate your lymph nodes during diagnosis? They're looking to see if they're swollen, okay? And if they're swollen, that means that the white blood cell population has really increased dramatically. That indicates infection. Lymphatic tissue is usually designated as two types, primary and secondary. Primary lymphatic tissue is where lymphocytes are born and educated. We'll talk later on about what education means. All lymphatic cells are born initially in the bone marrow. I'm sorry, all lymphocytes are born in the bone marrow. B lymphocytes are educated where they're born. They're homeschooled. T lymphocytes are born in the bone marrow, but they migrate then into another primary lymphatic organ where they are educated. So they're sent off for their education. Anybody know what that T organ is, where the T lymphocytes are educated? What this other primary lymphatic organ is, where the T cells are educated? T stands for thymus. Okay, so that's your other primary lymphoid tissue. We'll talk about that process later on. And we'll talk about what some of the secondary lymphoid tissues are. Things like your spleen and your tonsils and your appendix. I've got to go over to freshman registration, so I will carry it down on my way out to Natalie office.